Hey, hello. Welcome to this channel where I'm pleased to introduce Nebula and its physics-driven AI platform that enables structure-based drug design. So when you hear AI in drug discovery, evidently you acknowledge AlphaFold and its ability, thanks to data sets it considers, to predict most accurately structures of a majority of therapeutic targets. Nonetheless, today, 80% of disease-relevant targets are considered undruggable, and this is mainly because they present shallow protein surfaces with not well-defined pockets or hidden active sites. We have developed during the last 5 years at Nebula a unique generative AI algorithm that is based on reinforcement learning and physics-based modeling to leverage dynamic. That allows us, through this technology, to discover new conformations. Not only that, but also their population, the pathway in between, and the kinetics of transition. This is of paramount importance because it allows us to focus on conformations that have the largest population before getting the chance of finding first-in-class and best-in-class drug candidates. So our proprietary physics-based core technologies present several key advantages and allows Nebula to emerge today as unique in its value proposition. We do not need pre-existing set of data. Our conformations are provided with populations and discriminate between genetic mutations. Of course, our technology is cheaper and faster than experimental technologies, allowing to get some conformations, and we are agnostic to the type of therapeutic targets, which means that we can work on different therapeutic areas. So let me guide you to one of the many case studies to appreciate how our technology provides venue for discovery. In this case, we are interested in KRAS, a molecule that has been known for over 40 years as undruggable. If we consider starting only from an X-ray structure of one of the mutants, for instance, our conformation generated by our algorithm allows us to identify conformations with cryptic pockets, for instance, that are not known in the original structure. In this case, for instance, it's interesting because the pocket we discovered is identical to the one harboring a recently discovered inhibitor by Miriarty. Yet in another case, for another mutant that is implicated in a different disease, Nebula conformational Gen AI has allowed us to discover a cryptic pocket that is not yet identified by any other method. So today, next to this core technology that allows us to rank and prioritize druggable sites, we have developed several technological breaks to enrich our platform so we can really go from target to drug candidate with optimized ADMET profile. Evidently, our proof of concepts today are limited to small molecules, but we will be deployed to other modalities such as PROTACs and molecular glues. Hello again, I am Munir Tariq, the CEO of Nebula, with over 30 years experience in academic research in biophysics and physical chemistry. My co-founder is Daniel Vichev, with over 8 years of experience in AI. He's an expert in machine learning, and the team has been enlarged to include recently Fabrice Valavoine, who is our CEO, who has over 25 years of experience in drug development from lab to clinical trials. So through strategic partnerships, Nebula offers, as a tech bio, R&D services to partners as co-developers for their drug discovery programs. But Nebula can develop as well its own pipeline, choosing free patent space for lead candidates nomination. Today, Merck Digital Science Studio provides us with the opportunity to accelerate our development and expand our network by accessing both pharma companies and investors in the biggest market worldwide. So this leads me finally to conclude by thanking you for considering our application and looking forward to the next step of the process. Thank you very much.